Everybody. Thanks for tuning in to the offline viewing of our webinar, Interrogation of Biofilm Architecture in Static and BioFlux Flow Model Systems, illuminating environmental microniches and the dynamics of antimicrobial penetration. This webinar was presented by Professor Kim Hardy at Nottingham University. She's an associate professor there, as well as being a part of the National Biofilms Innovation Centre, UK. The talk will address a couple of important questions, specifically the difficulty in eradicating bacterial biofilms and new ways of mapping environmental microniches, in particular the use of novel nanosensors for pH. This biofilm model helps understand both the architecture and geography of the biofilms, and thus informs antimicrobial delivery and efficacy. The examples in this talk will include Pseudomonas aeruginosa, Staphylococcus, and Streptococcus mutants, as well as mono and polymicrobial biofilms on various surfaces. Without further ado, I'm going to segue into Dr. Hardy's talk, and I have to apologize, our recording of the webinar started late, so her talk will be starting shortly on slide 2. Thank you.
In fact, more bacteria are in biofilms than living freely in liquid. They're in the environment, in pipes like this picture, or also associated with hosts, for example, in the mouth, in wounds, or on indwelling devices like catheters. They start with a reversible attachment of the bacteria. So if they've got flagella or other motility mechanisms, they find themselves at a surface, and there they reversibly attach, and changes occur to allow them to irreversibly attach and become more strongly and intimately attached to the surface. And they may even move along using motility organelles like flagella if they've got them, or perhaps surfactants if they don't have flagella. Eventually they multiply and accumulate in small microcolonies, and there are some changes that are occurring where there's a matrix made, which includes the production of extracellular DNA and polysaccharides as well. And together these accumulate, and the cells again divide a bit more until you've got some quite large structures within a biofilm, and in between you'll have channels where the fluid can go. So in the end, they've got a complex coordinated community on a surface, and it's surrounded by a matrix of the polysaccharides and the extracellular DNA and proteins, as I mentioned. And within this, some of the cells will become dormant or change into persister cells, as they're called. And also there's a number of regulatory things that go on. For example, secretion machineries of proteins may be turned on or turned off, and flagellum may be turned on or off, depending on whether the bacteria want to move within the structure. So that's what's going on. And why are these difficult to eradicate? Well, because of this complex matrix around the bacteria, the antimicrobials that might be added from the outside of that find it difficult to penetrate through. And as they're penetrating through, they will come across different microenvironments. For example, nearer to the surface, there'll be less oxygen available, and there'll be more oxygen around the surface where you've got the channels passing by. As they move through, they might also induce stress responses in the cells, or these stress responses may be occurring because of these microenvironments. And one of those that's been documented quite well is the progression into persisters or dormant cells, which have a different physiology to the rest of the bacteria in the biofilm. So if we schematically represent the normal cells that are growing and dividing as green, persister dormant cells as blue, and also cells that will be within this and selected for that may be resistant to the antimicrobials that we might add. So if we have an antimicrobial that can kill the normal cells and the cells within the biofilm are not resistant to it, everything will die except for the persister cells, which will be dormant or changed in some way normally to make them a bit tolerant of the antimicrobial. But if the antimicrobial goes away and the conditions change, there may be regrowth, and then in which case you'd get regrowth of both types, the normal cell and also persister. If, on the other hand, your antimicrobial is able to kill this persister cell, but there are resident bacteria that are resistant to that antimicrobial, it would be left alive, and if conditions change, that would multiply, which would be a real problem, because not only would the biofilm be maintained in the environment, it would be even harder to eliminate, because it would be resistant to the agents that we have. So we want to study the formation of the biofilm and the structure of the biofilm so we can improve the eradication. So as a first stage, we can track antimicrobials as we add them to a biofilm, and we can also track the killing that occurs and which type of cell that may be killed as the antimicrobial goes through. If we're able to physically disrupt or use a chemical disruption technique to break up the outer matrix, it may be that we can improve the penetration of the antimicrobial and kill more of the normal cells. But if we were doing that, we would also want to map the environmental microniches so that we can see how far it's penetrating and what it's killing as it goes through. And if we were able to develop new or combination therapies that are able to target the dormant cells as well, it would be good for us to be able to track the cell physiology so we can tell that the dormant or persister cells are being killed. And it would be ultimately good to investigate new ways to kill the cells within the biofilm so we can also kill anything that might be resistant to our current antimicrobials. And so discovering new targets for antimicrobials within biofilms that provide such a way of killing those cells is another avenue that's worth studying. So on this slide, you can see another cartoon showing a biofilm with some bacteria in it. So if we're looking at the penetration of an antimicrobial, we want to know if it reaches only the surface ones or can get all the way to the middle. And then also, where will it be active? Will it be killing all the cells that are there or just some of them? So that's the level of tracking that we would like to achieve. And one of the tools that's helping us to is going to ultimately help us track the progress of antimicrobials are small optical nanosensors. This work is done in collaboration with John Aylott in the School of Pharmacy here at Nottingham, who makes these nanosensors, which are smaller than bacteria. They're made of polyacrylamide, and they can be charged or not charged, and they are containing different dyes. So there's one reference dye which is red, Tamra, and that is red all the time, so we can always find where the nanosensors are. And there are two other dyes, one's FAM and one's Oregon green. Both are green, but they are more green when the pH is higher. So that then gives us a gradient of colour, where if we've got more green, it's yellow because they're in the same place, and where the green is turned off, so it's a low pH, we see the colour red. And this has been quite useful for us. And the first way that we've tried to use them in the biofilm context is with biofilms of Pseudomonas aeruginosa. So this is a gram-negative bacterium, which is an opportunistic pathogen, and it's a particular problem for people who've got cystic fibrosis, or if you have a wound that's caused either by a burn or trauma, and also it causes eye infections. The types of infections it causes can be acute or chronic in their nature, And because it's quite a ubiquitous bacterium, there are many reservoirs all over in the environment which can serve to inoculate these kinds of sites. It's also a problem because it's very difficult to eradicate due to its intrinsic resistance to antimicrobials and the numerous virulence factors that it produces and the complex regulatory circuits that they are controlled by. So the first time we've added the biofilms, the nanosensors, is within static biofilms in an 8-chamber model. And we've incubated for 48 hours at 37 degrees and added the nanosensors. And if you view them from the top, we have a yellow color indicating a high pH. So if we just first test if they respond to pH by adding a low pH, 3% acetic acid, we can clearly see a lowering to the red color and the appearance of the red color. When we looked further at this natural pH, we could see over the 100 micron thickness of the biofilm a gradient from low pH to a higher pH, which was really astounding to us because we were hoping, but we weren't sure we were going to see such a good resolution in the pH gradient across the biofilm. And we were able to calibrate the pH from the colors that we could see with the two dyes and looking at the ratio of the two dyes and see that it ranged from almost 6 to about 3 across this 100 micrometers, which gave us what is going to be hopefully a very powerful tool to look at the microenvironments of the biofilm. But what we wanted to find out first was how fast the nanosensors can react to pH changes, because we don't always want to look at static changes like in this situation where we just added some acetic acid. So for that, we turned to a different bacterium using Streptococcus mutans, which this time is a gram-positive bacterium that's a late colonizer of dental plaque in the mouth. And because it can create acid production, which demineralizes the tooth enamel, this leads to the tooth decay that you can see here. So what we were pleased to see is when we added the nanosensors to this and biofilm of these, again in the chamber model, and then added a succession of different buffers at different pHs, we saw a resultant change in the pH. So this is pH 6, and we can see the yellow color. pH 5 we step down, and then pH 4 we step down again, and visually you can see the orange color as the red is more obvious, and then we stepped back up to pH 7 and we returned to the yellow color we saw at the beginning. So this is really good because it shows us that within a very short amount of time, within a minute, the pH solution has penetrated the biofilm and the nanosensors have responded. So we wanted to see if we could see this response within the flow biofilm model. So this is where we employed the BioFlux nano chambers, and we pumped fresh media constantly across the biofilm. And these, you can see here in the picture on the left, you get a manifold going into the plate where you can flow the media across, and the channels are designed so that you get a uniform shear across the whole of the channel, so you can have uniform pictures of your biofilm. And because we can do time-lapse and Z-stacks, we're able to make little videos of these, which are really useful for the analysis. So the actual setup involves an adapted microtiter plate, and you have an inlet and an outlet with the channel in between. The top surface is made of polystyrene, and the bottom surface, which is where you've got the viewing objective, is made of glass normally, but Fluxion are also developing a removable PDMS version, which might be useful for different analysis down the line. The plates come in two different forms, either 24 well or 48 well. The 48 well is the simplest, with one inlet and one outlet and the channel in between. The 24 well plates are more complex because they have 2 inlets and 1 outlet, with all the channels being together here in the center of the well. From the inlet, you have a serpentine, which leads into the actual channel, which then goes directly to the outlet. And the advantage of the serpentine is that you don't get contamination from the channel to your inlet well. So here's one of the videos, which has just started here on the left, and you can see it's yellow, and the nanosensors are there. So initially we're seeing yellow, and then as the microcolonies start to grow, you can now see, I hope, some spots appearing, which are getting progressively bigger, and the green colours are beginning to be seen. And if you look very closely downstream of some of these colonies, you can see a comet tail of red, and that is because there's a local pH change that is causing the nanosensors to show up the low pH locally. So in close up, you can see here the comet tail downstream of microcolonies, which we think is because we're growing, in this case, on succinate. So it's used up, the pH downstream will be used up, will change. So now we can see pH changing in channels. What we would like to know is what happens to the pH during an antimicrobial penetration. Antimicrobials also penetrating, which we're hoping we might be able to see have an antimicrobial with a distinctive pH, and also how these antimicrobials actually kill as they're penetrating through. So I'm going to go through first a population of the antimicrobials and the killing to be tracked over time. So for this, we're using different tools. Here you're going to see a video of a microcolony inside the BioFlux growing, producing GFP, and you can see it gets greener and greener and greener, showing it's alive, and then that recedes because it starts dying. And on the graph, which should come up now, you can see the scan of that, where the center of the microcolony is getting greener and greener and greener and dying. In parallel, we also measured sorry, I'm trying to start the video, the propidium iodide uptake, which goes into dead cells, and you can see an outer rim first, and then the center becomes red. But unlike the green, it stays red. So here you've got the scan. You can start to see the 2 shoulders appearing, and then the center gets redder and redder and redder, and then it stays red as the cells are all dead because the nutrients have been used up, presumably. So going on then one step. I can get the picture. Sorry, the slide to move. Okay, so again, using the BioFlux, we wanted to move now to the 24 well plate, because this enables us to switch the media in the inlet, so from one inlet to another, and this allows us to add an antibiotic at a certain time, and then follow the transient introduction of that antibiotic through the channel. So here you can see 2 different concentrations of ciprofloxacin that have been added to our biofilms of Pseudomonas aeruginosa. And you can see the redness in the one on the left is slightly less than that in the one on the right. Again, we're looking at propidium iodide being dead cells, showing that the higher concentration of the ciprofloxacin is killing more of the cells. And we were able to track that over time. So we added our ciprofloxacin at 16 hours, and then within 2 hours, it was the appearance of the red color, the propidium iodide, showing that the cells were beginning to die. And you can see that here in these 2 videos running side by side. Here you've got the propidium iodide. When the asterisk appears, that's when we add the ciprofloxacin. There it is. and then a couple of hours later, all the cells are dead. Just to show it, that it's not just this antibiotic that does the same trick. Here is tobramycin, and again we're watching green alive and red dead cells, but this time instead of GFP we've got cyto9, and as time goes on you'll start to see some microcolonies appearing, which is happening now, and then suddenly the red starts to appear, and in a moment you can really clearly see that the cells have died. To show you one more example, I'm going to show you benzalkonium, because it, as well as killing the cells, it can also work as a surfactant. Again, we've got cyto9 and propidium iodide staining of live and dead cells, and you'll see in the channel that the green cells are growing well, and then they start to die. It's slightly out of focus on this occasion, but you can clearly see that at the end of the treatment that the cells biofilm is sloughing off and being flown away to the outlet tube. One other thing that we like about the two channels is the ability to add at the same time media to one and media plus antimicrobial to the other, and that creates a gradient across the channel. And we've done this with ciprofloxacin and Pseudomonas, and you can see here that at the bottom there's more depth because we're measuring propidium iodide, where the concentration of the antimicrobial is higher. And over time, as you can see in this revolving video, that depth spreads to the top of the channel. So that's all well and good to look at the simplest types of biofilms, but nature is a bit more complicated. So we've also tried to see how well this works in polymicrobial situations. And the first one I'm going to show you is where we've put some commensals, which includes Staphylococcus epidermidis, a gram-positive, and Micrococcus luteus, also gram-positive, which promote skin health. And when they're perturbed, you can also get initiation of autoimmune diseases. And we've paired those up with pathogens, including the Pseudomonas that I've talked about before, and also Staphylococcus aureus, which is another gram-positive. It's both a human commensal and also a human pathogen, and it can form biofilms, and is a particular problem in hospital infections, and especially ones associated with indwelling devices. And one of the reasons we were interested in Staphylococcus aureus is because it and Pseudomonas aeruginosa co-infect a number of different sites, including wounds and cystic fibrosis lungs. And there seems to be some sort of interrelationship between the ratio that's there and the prognosis for the patient. So here you can see some of our biofilms that we've grown on top of skin cells, keratocytes, which we stained up with cell masks in purple. And you can see if we have just the pathogen, there's a lot of damage to the eukaryotic cells. With a low number of the pathogen and with commensals, there's a little bit of damage, and with a high number of the commensals, the pathogen with the commensals, there's much more damage because the cells are starting to look broken. And then if we look at the biofilms that we've put on top of these cells, so here you can see Pseudomonas in red on its own, making a nice biofilm on top of the eukaryotic cells. If we have low numbers of the Pseudomonas with our commensals, and we've stained up the staph epi in green here, you see two layers, which is what we think is protecting the eukaryotic cells from damage. Because if you have the higher amount of the pathogen, this layer is getting breached, and we suspect that's what's occurring to allow the pathogen through to damage the eukaryotic cells. We've also seen similar changes, similar damage when we put Pseudomonas on with Staph aureus. What's interesting here is that with the Staph aureus, you never get these two layers. You always get this punctate arrangement of the two pathogens and a lot of damage to the eukaryotic cells. So moving on from that to the more complicated biofilm model where you're adding more than one antimicrobial, we've decided to have a look, to follow up some work that's been published on E. coli, where pre-treatment with the antimicrobial triclosan, which is used in many household products and inhibits fatty acids synthesis, somehow seems to give tolerance against the antibiotic ciprofloxacin, which we were using earlier. So you can see here in this CFU counts from broth cultures that where you've got triclosan, more cells are surviving than where you haven't got triclosan. And the longer you incubate, the more obvious that becomes. So what we wanted to do was try and see if this also was true in biofilms. We chose to do it with Staph aureus rather than the E. coli. So the first thing we did was to see what happened in a broth. So with increasing amounts of triclosan, we see increasing amounts of death of the Staph aureus, which made sense. But because Dean also added the pH nanosensors, which we introduced at the beginning, he was able to monitor the pH. And what he could see was a large pH change in cultures with low amounts of triclosan, and you only saw a small amount of that change in the pH when you added a lot of triclosan. So then we came up with the theory that perhaps this lower change in pH could reflect a slower metabolism of the cell. And when you've got slower growth, you would have less activity of the target of the antimicrobial, the ciprofloxacin. And that means that the cells would be less susceptible to the ciprofloxacin. So we looked first in the broth to repeat the experiment I showed earlier with the E. coli, and you can see that when we pre-treat with the triclosan, we have less surviving cells. When we've then added the ciprofloxacin, and you can see here as well, this is cells treated with triclosan and ciprofloxacin surviving, whereas on ciprofloxacin alone can kill them off quite easily. And to see what's happening in a biofilm, that gave us the impetus to have a look in there. And when we looked in our Staph aureus biofilms, again, we've got the nanosensors there to have a look to see what the pH is. And although I'm not going to talk about the pH, what our nanosensors were able to show us was that upon treatment with the triclosan, we have a much thicker biofilm, and there seems to be this surface structure above that is stained up by the nanosensors. Here you can see that the bacteria are in blue because they're stained with DAPI. So there we've got an effect of, and we're able to monitor it between one antimicrobial and another, but we haven't really shown you how we're seeing where the actual ciprofloxacin is going. So in some other work, we've been trying to see if we can measure where that goes. So first of all, looking at biofilms grown in chambers, we looked at Staph aureus and added ciprofloxacin, and you can see the death of the cells closer to the surface where you've added the ciprofloxacin. But in order to measure where the ciprofloxacin are, we had to increase some of our technical skills and tools, and we used mass spectroscopy to see if we could find the ciprofloxacin. So in order to do this, we started off by drying onto a glass cover slip either ciprofloxacin or ciprofloxacin plus bacteria, and having a look in the mass spec, the TOF sims, to see if we could find the ciprofloxacin. We could easily see it alone, and in the bacteria we could also see and detect it. And that was dependent on the concentration. So at the lower concentration, we saw less, and at the higher concentration we saw more. And to see if this was possible also in a biofilm, we grew biofilms on glass cover slips, and this is the baseline data from the mass spec. So this is actually where the glass is. This is where the organic material is, and there are areas here which are high for the organic material but not for the glass. So that shows us where the ciprofloxacin is, and we could detect that within a microcolony, which we think has got a lot of scope now as the mass spec technological platforms improve using nanosims and orbisims. We should be able to detect this even better, and also with collaborators on our EU project, we'll be able to extend that to other techniques to look at the metabolites and the antimicrobials, for example, vibrational spectroscopy like Raman, super resolution microscopy to see the cells even better, and also X-ray fluorescence and measurement tools, which will help us to track the antimicrobial alongside the death of the bacteria through the biofilms. And so what I hope I've shown you in this short presentation is how optical nanosensors can facilitate mapping of the environmental niche, micro niches of biofilms, how the BioFlux flow models can be used to interrogate the biofilm architecture and geography, and how we can track antimicrobial penetration and the killing of the cells that results from that. We can do this in polymicrobial complex communities and also when we combine different antimicrobials. And we're at the beginning of looking at some new technological platforms that the resolution is being improved of all the time so that we can track everything in much more resolution. But there's still more to know. We would like to be able to link all of our findings to the extra polysaccharide and the other matrix components and virulence factors, and to see if we can manipulate the polymicrobial biofilms to make them easier to eradicate or not form in the first place, or for antimicrobials to penetrate them better, and to see if we can make the antimicrobials more efficient, because if we understand the geography and architecture of the microniches, perhaps we can deliver them better. And looking at the dynamics as also of the penetration will help us to find novel delivery strategies to improve our eradication of biofilms. And lastly, I'd like to thank all the guys in the lab. You've seen work from Dean, Eleanor, James, Birta, Mark in this presentation, and all along the way I've mentioned who I've collaborated with and the people who've generated the data. So I'd like to thank you all for listening and see if there's any questions. Thank you.
Thanks, Dr. Hardy. This is Christian from Fluxion.
Yep.
To all the attendees, if you do have some questions, I would encourage you to type them into the interface and we'll read them aloud. I did have one question that I wanted to ask, which is, you've recently got these assays up and running. What was the most viable, reproducible biofilm flow assay?
It's very strain dependent. So on some cases, the organism's best in the chambers, and sometimes it has to be quite carefully optimized in other systems. It seems to be very critical what the media is. And you could also get some variation depending on the strain. So Pseudomonas PA01 and PA14, which are two often used strains, seem to have very different requirements when you're trying to find a reliable model to look at them.
Okay, thank you for that. Yeah, I think getting those models tuned is very important, and we will, as we get more and more people doing similar experiments in similar systems, I think the hope is we'll get to a body of knowledge of how to reliably get all these organisms to grow and get to the biofilm state. Just trying to see if there's a couple of questions coming in. One question is regarding the poly-M keratin model, were the two organisms seeded at the same time?
So we put the commensals on first, and then we put the pathogen on later, because that's the nature of the way that we want to do the experiments, to find out if the commensals can be protective. But if you wanted, because we put more than one commensal on at the same time, you can also do it in the way where you would add more than one bacteria at the same time.
Okay, thank you for that. And I just want to encourage you, if you're asking questions, I'm sorry, the interface is not so great with follow-up questions, but if you do have one, type it in and I promise I'll get to it. Hopefully we're answering the questions satisfactorily. Another question is, how do you expect the different architectures, multi-layered cells versus honeycomb-like structures, to affect the antimicrobial penetration?
Sorry, which I missed the middle of that. The what versus what?
The question is the multi-layer cells versus honeycomb-like structures. The different architect. The question relates to the.
Oh yeah, okay.
How do different architectures affect that antimicrobial penetration?
So I think that it depends mostly on I guess the way that the matrix is built up, because the diffusion between different matrix components, which will be different depending on the bacteria you're looking at, will affect the microenvironments, because it will affect anything diffusing from the bacteria out and also anything diffusing from the environment in. And it would also probably affect the channels, kind of where they are, how fast they're flowing, and those kinds of things. So you would have differences in things like oxygen and water availability to cells, which I think would affect how many cells die and where they die. And as I said before, the penetration of the antimicrobials through, where they can actually get to, and also where they might be active. Because if, for example, there's a big pH change and your antimicrobial is pH sensitive, it will work on cells next to a certain pH, but not cells next to another pH. Did that make sense?
Okay, thank you. The next question is, were you able to gain additional three-dimensional information in biofilm structure along the z-axis by using MALDI?
Not yet, no. Yeah. So we're still in the stages of optimizing the mass spec analysis of the biofilms. So yeah.
Okay. Next question relates to this is something I was going to ask as well. What are your thoughts now, based on the experiments to date, on the static versus flow conditions in terms of the differences you see?
Well, I think all I can say is that there are differences between the two systems, and I think you need to use the model where you can answer the question that you have, whether it's if your bacterium's more reproducible in a static one, then it's better to use a static one. If it's more reproducible in the biofilm that it forms in the flow, then it's better to use a flow. Because I think what's really crucial is having that reproducibility, because if you don't have reproducibility, you can't tell the difference between two different treatments or two different strains or something like that. And then, as well as that, you've got to think of the question you're answering. So sometimes the experiment that you want to do can't be done in a flow or can't be done in a static system.
Got it. And a follow-up to that, I guess the introduction of compounds is while observing the biofilm has to be done under flow, but the follow-up question to that was, were there clear differences in biofilm architecture or drug penetrance between the two systems?
I don't think we've done enough analysis to say one way or the other. I think you can certainly see that there are the same kind of gradients available, but they'll be in different places. So I'm trying to think of an example. I think that the gradient of the pH that we see across biofilm in the static model that I showed initially is a bit different to the one in the flow where you have a comet tail of pH coming afterwards. So there are some differences, but I don't think we can say what those differences are and what the importance is of what they are at the moment. I think we need to do further study, but that's a really important point for us to bear in mind, that we try to actually compare across and make sure what's the same and what's different.
Okay, and the next question is, have you studied the pH gradients in biofilms within the BioFlux? And are there any experiments that you're aware of that also look at metabolic gradients? Because those are important in antibiotic treatment efficacy.
I think there are experiments where they're looking at metabolic changes in response to antimicrobials. There are limitations on the technology platforms, so they're quite the studies are quite confined at the moment. So there's a lot more that needs to be done to understand. And one of the things, for example, is in the BioFlux, with it being a closed system, it's difficult to take part of it out and analyze it on a different platform. So that's why having the new removable bottom side is very useful. And the other part, I think they're going to have reusable small windows that can come in and out, which you can put your own substrate in. So that's going to take that forward, but those sorts of things have not been available for long enough to answer that question. And where you've got other systems, other flow systems, where you can put coupons into a flow and take them out, there are some work done with those, but it's more the resolution of the platform that you've been putting them onto is limiting. So the sizes we're talking about are very, very small, and so you need greater resolution to see the metabolism inside one cell next to what the metabolism is inside the cell next to it in the biofilm, next to what's actually in the matrix around it. Did that answer the question?
And I hope so. Unfortunately, it's hard in this to get, but we do. There are now available plates with a access window where the end of the experiment, so that should. I'll just go through a couple more questions, even though there are more, just in the interest of time, since we're approaching the hour. One question is, does the BioFlux allow for bacteria being introduced onto preformed biofilms from one inlet, and in parallel to the second inlet, which would have medium at set time points? And I think this question refers to, can you form a biofilm using the 24-well plate and then introduce a different type of bacteria at a preset point? And I've got my own ideas as to that, but would like to hear your answer, Dr. Hardy, first.
We normally seed from the other end. So we seed from the outlet in so that we don't get growth into the serpentine. What I'm not sure about is whether a bacteria would fit through the serpentine part, which perhaps you can answer.
Right. So bacteria definitely fit through all the different parts of the plate. The channels are 75 microns high throughout. I think, yeah, one can envision experiments like these, exactly how to do them and not get contaminate the media inlet is more complicated because the outlet for that reason. There would probably be a possibility of flowing in the opposite direction in the 24-well plate. In other words, having one inlet and two outlets instead of two inlets and one outlet, and then coming back and seeding from one of the outlets. That's possible. I think one could certainly introduce inoculum at a certain point and have those cells interact with the biofilm. I think that if you introduce inoculum from the inlet, again, you wouldn't be able to do that for very long, just because if you contaminate the inlet, that's going to lead to problems very quickly. So I think it just depends on, the type of experiments that's envisioned. I think for a short-term experiment, you could certainly do it. Contamination is not as much of a worry there, but I think to do this for longer-term experiments, it would take some real, some method development. Okay, we'll take one final question. I think then we'll adjourn. And one final question is, does the BioFlux allow you to test the effect of multi-compound cocktails and biofilm resistance, and do you plan on doing that?
So more than one antimicrobial? Well, you could certainly add more than one antimicrobial. And yes, we are planning to look at antimicrobial resistance, but we'll probably do that using mutant strains that have a variation in their antimicrobial resistance, and then see how they respond when the antimicrobials are added. One of the things that makes that quite challenging is sometimes these mutants that are more resistant to antimicrobials are also less able to make biofilms. So that sort of gives you another technical issue that you've got to get around.
Yeah, and I think that there certainly have been studies in the past that have already been done where I've seen different concentrations of compounds and as well as combination of different compounds being introduced and looked at in the BioFlux. And I think what people do there is they just pipette in the let's say 2 compound combination into the inlet, and then flow it over the biofilm. So I think that's something that's fairly straightforward to do. All right, I think we're gonna adjourn here. Most people are on their way. Thanks a lot to everybody who attended. If you questions that weren't answered, which there are maybe a couple, we will make sure to answer those offline. And if you missed the webinar or some of your colleagues want to see it, we will have a recording available on our website. And to finish, I'd like to thank again Dr. Hardy and wish her the best of luck with the research. And yeah, thanks to everyone for attending.
Thank you.